My main goal during the session is really to help other both researchers as well as clinical practitioners within the field to understand what are the fundamentals of proteomics. And so really what we want to talk about is how we can use mass spectrometry-based approaches for the most part. And this is a powerful instrument that we and many other labs around the world have that let us evaluate thousands of proteins within either myeloma tumor cells or immune cells within the microenvironment, helping to understand mechanisms of either myeloma progression, myeloma development, drug response, drug resistance. Those are some of the techniques that we've looked at the most, or some of the biological processes that we've looked in our lab. But really trying to say, what's the power of this? How can this help us understand myeloma? We know there's been so much research done in the field on myeloma genomics, myeloma transcriptomics, looking at mRNA expression. But in this context, the mantra of my lab is that biology happens at the protein level. And so the idea here is that can we get to that level of actually understanding how proteins are changing throughout the disease course in response to drugs, and then looking at aspects of myeloma biology you cannot see just by doing genomics or transcriptomics, specifically how proteins are changing their localization within the cell, how protein-protein interactions are changing, and how that are going to impact drug response, for example. And so these are kind of the techniques that are now emerging within the proteomics field. And one thing actually we'll talk about as well, and we're not doing this yet in my lab, but there are emerging approaches for even single-cell proteomics using mass spectrometry, trying to get down to the same coverage that we have now with all the single-cell approaches that we've seen at this meeting have really exploded in myeloma, and I think are really going to make a big impact. Can we take that to the proteomic level? And so that's really what's coming next within proteomics. And so I think it's being able to introduce some of those techniques, capabilities, technologies to the broader myeloma field is my goal with this session.